Nanfang Hospital is the first affiliated hospital belong to Southern Medical University. It's located in Guangzhou, South China. Originally, the first affiliated hospital of the first military medical university, which founded in 1941. So after 80 years of development, it has become a comprehensive hospital with around 3,500 beds. So a big hospital. And this hospital integrates this special zone innovation and the culture of Red Army. And I previously served as the executive chair of Education Committee of ISEF from 2020 to 2022. And we also established the Chinese Society for Extraterrestrial Vesicle CSEV in 2017 in Guangzhou. And I'm now the position of the Vice President of CSEV. So I will give a brief introduction about the EV biomarker technology. So these three parts. First of all, we'll give a brief introduction about EV biomarker. As you know, extracellular vesicles are nanovesicles are released by cells to extracellular environment, which carry various biomolecules such as protein, nucleic acid, and lipids, etc. The EV is involved in regulating various physiology and pathological processes, such as like in many human disease. Currently, research on EV is flourishes, especially in the field of EV biomarker. You can notice that more and more company is growing based in EV research field. And a variety of specific bioactive molecules enriched in EV, such as protein, nucleic acid, metabolites. All of these biomolecules hold the potential to become novel targets for disease diagnosis. In different diseases, EV biomarkers exhibit diverse types. For example, the PD-L1 positive EV has the promising application as a predictive indicator of effectiveness of PD-1L1 therapy in cancer. Additionally, the detection tools for the early hepatocellular carcinoma, HCC, are not ideal, and the abundance of small RNAs in EVs can provide a new approach for early diagnosis of HCC. So, as for the EV biomarkers, we need new technologies to detect this biomarker from discovery to clinical application. And this includes techniques for isolating EVs from our body fluids, different kinds of body fluids, as well as technologies for screening biomarkers based on multi-omics approaches. And we also need to validate these candidate biomarkers. As for the EV isolation, they commonly used method including the ultracentrifuge, density, gradient centrifugation, size, exclusion, chromography, etc. And we also noticed that there are a series of advanced nanotechnologies have been developed for isolation of EVs, which can be categorized into three board types. The separation based on physical features, chemical affinity on microstructures, and the nanostructure chemical combination. For example, technique like the thermophoresis, it employs temperature gradients to enrich and isolate EV. And EV also can be isolated titanium oxide based on high affinity toward phosphate. In addition, the involvement of proper nanostructure surfaces in EV isolation have been reported, which could not only provide a higher efficient reaction rate, but also overcome the limit to interfacial EV bindings. So as the EV biomarker screening strategy, the multi-omics biomarker screening direction mainly include proteomics, post-translational modification proteomics, metabolic omics, and transcriptomics or microbiomics based on high throughput sequencing. For example, proteomics, matter biomarkers studies typically use the mass spectrometry technology, and the transcriptomics research is mainly based on RNA sequencing technologies. So with the new continuous innovation of new technologies, these technologies are evolving. New biomarker screening technologies are emerging, such as the proxy extension assay, reverse phage protein array. And after screening, we need to verification these EV biomarkers. There have been many new analytical methods being developed for EV biomarker verification. They can roughly divide it into categories: label-free and label strategy. And the sensitivity, specificity, and repeatability have been improved a lot during these years. The label-free strategy is developed based on the physical characteristics of EV, including Raman spectroscopy and optical tweezers owing to high sensitivity. And the label strategy may include two key processes. Firstly, the protein or nucleic acid would be recognized especially by antibody, aptamer, or molecular beacon. And then it would be coupled with the CRISPR or PCR system for signal amplification and analyzed in microscopy or fluorescence measurement platforms. And these techniques shares the merits of high sensitivity and high specificity. And as this single EV analysis, we noticed that with the rapid innovation of EV detection technologies, and the EV verification has developed from the bulk analysis to single particle precision analysis. There are increasing demand of developing more sensitive, specific EV detection system based on a microscope, flow cytometry, or microfluidics. Additionally, a new TUF system was developed in which only 90 microliterature samples were needed for simultaneous detection of protein and nucleic acid like microRNA, messenger RNA in a single EV. within just 6 hours. Another one promising single EV analysis platform is high-sensitive nano-flow cytometry. It can detect 24 nanometer low refractive index nanoparticles, which has breaking the limitation of the traditional flow cytometry. By labeling the EV with fluorescence antibody aptamer, it could be used of single EV protein analysis. And we also developed a new microfluidic platform for EV subpopulation analysis at single EV level, EXO-ELISA. It could be used for averagely quantitative detection of single EV based on this membrane protein, and is around 10,000 times better than the ELISA. Okay, the third one I'd like to introduce the clinical trial and the regulation considerations. In our search in clinical trials for clinical trials related to exome or EV, we found appropriate 485 clinical studies related to EVs. As shown in the graph on the right, the tumor EV markers represent a significant proportion, which circulating and urinary EV also be highly researched area. A number of liquid biopsy products based on exome marker diagnosis are currently available on the market, including like the ExoDiagnosis DX Prostate, which detects 3 EV microRNAs in urine samples, correlated with the degree of risk of prostate cancer. And in this year, China also announced the first exome diagnosis products approved for marketing. The human exome CA125, HE4, and C5A test kit, which used to detect the human serous exosomes in vitro for diagnosis, the epithelial ovarian cancer. This kit had good sensitivity and specificity for different stages of EOC. And when applying for FDA certification and other approvals, researchers need to consider several factors: the clinical validation and the efficiency assessment, and the manufacturing process quality control. And there's also a number of considerations, including documentation and record management, clinical trial design and execution, application file preparation, and continuous oversight and improvement. Okay, of course, there's a challenge and perspective. There's still an urgent need for more comprehensive technologies for isolation, verification, and analysis of EV, which is suitable for clinical laboratories. There's till now a lot of basic research, but very few can translate to clinical application. So they also need such as clinical large-scale population validation, different population disparities, and some challenges in clinical translation regulation. So the field of EV biomarker involved multidisciplinary intersection, including molecular biology, basic physiology, biostatics, technologies, and machine learning, etc. So we need to more collaboration with interdisciplinary scientists. So we have established interdisciplinary medicine journal. We want to improve and report these research results based on the multiple disciplinary medicine. Okay, thank you all for your attention. Thank you.
Thanks, Larry, for the very nice, I think, introductory and summary talk, and it covers everything in this field. If you are developing like a new method, new technology for EV using liquid biopsy, I think he's the right person to talk to. And he will be there in the Meet the Expert session later this afternoon, so please feel free to talk to him. Is there any questions? I think we have time for one or two questions. If no questions, let's introduce the next speaker, Andrew Lai from University of Queensland, who will be talking about detection of multi-cancer signatures from EVs using automated HPLC.
Hello, it's great to see so many EV enthusiasts here under one roof. I would like to thank the IOC for organizing such a great event. So my talk is about using high-performance liquid chromatography, HPLC, to automate EV enrichment from biological fluids such as plasma. So my talk is about, broken down into several bits. First is about my rationale of why using HPLC is an ideal platform for EV enrichment. Next, I'll talk about some method development, in particular focusing on feasibility and reproducibility. And finally, I want to talk about using this method and apply it as a proof of concept to separate EVs from different cancers. So why is HPLC a good platform? First of all, HPLC can be ubiquitously found in pathology labs. Therefore, any diagnostic test can be readily slot into the existing network. Also, HPLC is automatable. As you know, humans are not the best in doing repeatable things. So HPLC can reduce manual errors. And also, HPLC is modular as well. So depending on your needs, such as if you want high throughput, you can add multiple columns to the single HPLC. It is also adaptable, so the enrichment of EVs can use different chemistries, such as the ever popular SEC, the charge-based IEC, affinity. And for this project, we use a hybrid capital core, so which is a hybrid between charge-based and size, and which I think is can separate EVs more. So, so the HPLC purification cycle. So you have the sample in autosampler that gets injected into the column. Fractions were collected, so the EV and non-EV fractions. Next, you want to remove the bound material from the column and finally equilibrate for the next column. Initially, just as a test of concept, just to see if it works, I inject FBS, which is common and cheap. However, next we went to the clinical sample. So for reproducibility studies, we obtained 4 plasma samples, and we inject each plasma sample 3 times in triplicate, and each time we obtain 7 fractions each. So, as you can see, most particles can be found in fractions 1 and 2. In addition, you can see that the technical replicates within each biological are similar. Also, we think that potentially the EVs are in fraction 1 and 2, and with fraction 3 and 4 as the non-EV fractions. So plotting against TSG 101, the CD81 shows that it's reactive in fraction 1 and 2, and also the fraction 1 and 2 are reactive with consistent EV morphology. So now we know this method works. Can we apply it to a larger cohort? So no, we don't. We haven't shown that yet. So we need to show it's reproducible first. So from a technical injection, we can calculate the coefficient of variation. So based on this, we can see that the inter-CV of the particle size and mode are very low, around 10% or lower. In terms of protein, the CV was around 8.5%. So thus, this method is highly reproducible in terms of particles obtained and the protein yield. So now I can say that the technique is reproducible. So can we apply it to a larger cohort? So what we got was we obtained 148 different plasma samples across 5 cancers. For each cancer, we inject each sample twice, consists of 100 microliters of plasma. So the EVs coming from 100 were used for RNA extraction, and the other 100 were used for protein lysis. More importantly, a pooled plasma was used as control and was injected every 10 injections. The EVs enriched was analyzed with downstream analysis, including small RNA sequencing and MS proteomics. So as I mentioned, it is important for large cohort to ensure that the separation, in particular the capital core, is separating the EVs and also is binding to the non-EVs component. What we can see in the EV fraction is that the protein contained in this fraction did not go up that much. In addition, the protein contaminant that is bound to the column did not reduce, thus showing that the capital core can still bind to the non-EV proteins over the 90 injections. Similar trend could be observed in the particles. We can see there's not much degradation in terms of particle number. So next, so we did mass spectrometry on multiple cancers. So you can see got ovarian, brain, lung, colon, and endometrial, and the control for benign normal. So we obtained protein profiles of each and did a pairwise comparison against for the cancers versus the normal. So we obtained proteins that were upregulated and downregulated for each. And for example, for brain metastasis, we obtained 121 proteins up and 196 proteins down. That's confirming that our end-to-end workflow can be used as a biomarker detection. We did similar work for small RNA. So we did comparison. We also identify microRNA that were upregulated and downregulated for the cancers versus controls. So ultimately, we now have 2 different data sets. So ultimately, what we want is to generate a classification model combining the 2. So what we did was to use the mRNA and the protein into a Python package. And with this package, it ranks different classification models to separate between the cancers and non-cancers. So here, the row curve showing that cross-validation, we can separate the different groups at over 90% efficiency. So in summary, I gave like a small, I gave some reasons why HPLC is an ideal platform for automated enrichment. I gave some data showing that the utility of HPLC combined capital core in the EV enrichment with, that is compatible with the downstream analysis. that can perhaps be translatable into pathology lab. So the inclusion of the QC can give information about a column and thus gives you improved reproducibility and record tracking. And I'll show you a little bit of data about the generation of different classification. So I'd like to give a quick shout out to all the group members and all the posters and the talks they're going to give. So yeah, just ask them questions. I would like to acknowledge collaborators and so in particular clinicians who gave us samples. And finally, I would like, so the University of Queensland just recently started the UQ Centre of Nanomedicine, and so it's a national international network. So yeah, gaps then, I suppose.
Really lovely presented data. I was wondering, you showed your QC for the HPLC.
Yep.
Did you assess the reproducibility in your omics data? Like, for example, with those technical repeats, did you see them clustering really closely when you did like a principal component analysis, or did you see like technical variation over time based on the HPLC?
That's an excellent question, and I would add it to my list to do. Yeah, that is something really good. So we should be doing it.
Yeah,
absolutely.
I mean, you've got clearly a really strong signal anyway, so that's not like an issue.
That's a really good, good idea. Thank you.
Hi, thanks for this great talk. My question is, what do you think about for the next, if you want to transfer HPLC in clinic, do you think it's easy or not? What do you, how ideal, what's happening from that?
Well, easy is a relative question, right? So how much resources you willing? Certainly, any assays, I feel that you isolate EVs using HPLC system is built. Your assay needs to come from there, right? Because each isolation technique will have its own unique subpopulation. So I feel like if you want to create a new assay, you need to start with the HPLC system and work downwards. I think it's much harder to translate EVs isolating different techniques and then bring in HPLC.
That was a great talk. Look, when it comes to pinpointing disease, particularly in circulation, disease-specific proteins or disease-associated proteins tend to be 1,000-fold lesser than abundant components in plasma. With that in mind, could you please give us an idea of what the protein coverage was for your EVs? And the nature of the kind of proteins you identified in your mass spec data.
Yes, the mass spec data. So separation of EVs from plasma is always very, very hard. So as most of you know, the plasma abundant proteins makes up a large, vast, what you can see. So bring in mind that in terms of biomarker, we just want to separate between our case and control. So even though, yes, it would be great if we deplete all the high abundant stuff, but however, if we can separate from what we can see, then I think it's good enough for me, for us, or for you. However, not to say that we should not be trying to achieve a high, more highly enriched sample. But I think there's no like the absolute gold standard yet in terms of speed, in terms of all the different. There's always a plus and minus for each.
Thank you.
Let's check Andrew again. So next will be only two rapid file over top because it does teach certain theory couldn't join us. So I'll let my colleague Lei introduce the next speaker.
Okay, the entry that talk. And the first speaker is Abba Kumari from University of Michigan. And his topic is co-optical detection and mutation analysis of cancer-associated HUS. Please.
That topic is actually different from what I'm presenting. There was a mistake in the website. Yeah, no worries. Hi everyone, I'm Abha, PhD student in chemical engineering at the University of Michigan. I'll be talking about using exosomes in glioblastoma patients, especially in the context of opening the blood-brain barrier. So GBM is a deadly tumor of the brain with a high recurrence rate, and detection of progression or pseudo-progression is very difficult because of the tight endothelial junctions in the tight junction in the endothelial cells which make up the blood-brain barrier. And scientists have looked into opening the blood-brain barrier Scientists have looked into opening the blood-brain barrier by injecting microbubbles and sonicating them so they loosen the endothelial junctions and physically open the blood-brain barrier. Now, using a device like SonoCloud 9 and pulsing that with low-intensity pulsed ultrasound has actually shown to increase drug penetration into the brain. But this has been studied in this context. However, I would like to leverage the opening of the blood-brain barrier to gather more tumor information in the peripheral blood and isolate exosomes, especially in my context. So the way this study was designed was in collaboration with surgeons in Northwestern University, and what we do is we collect a number of blood samples, starting from screening sample to some samples collected during the surgery called intraoperative samples, some collected after surgery during chemotherapy cycles, and two samples per cycle are collected, one before sonication, one after sonication, and then I isolate these exosomes from the plasma that is collected from the blood. The way I isolate exosomes is using an immunoaffinity-based microfluidic device, and it is based on the understanding that based on the observation and studies that phosphatidylserine is overexpressed on the surface of cancer-associated exosomes. And to target that, I use an XN5. So I coat an XN5 on a glass PDMS microfluidic device, and when I flow these cancer-associated exosomes, they are bound on the device in the presence of calcium ions. The beauty of this chemistry is I can release these exosomes using calcium chelation by EDTA and then use them for downstream applications. I tested this with first cell line derived EVs, and then I was able to capture them on the chip, release them, and this is a typical NTA plot that I get from the released EVs, which shows the EVs are in the exosomal size range. Then I compared NXN5's performance with other chemistries like EGFR and CD63, and I found that NXN5 would perform best when compared to these two, which is shown by NTA, capture efficiency, and DIO staining as well. Once this was successful with cell line derived EVs, I tested this device and the chemistry with plasma of the patients, and I found that NXN5 again performed better in terms of protein quantity that I obtained from these samples. So we can see that CD63 was not able to distinguish between patient versus healthy, but NXN5 was able to do that. Then I looked at some protein markers, exosomal marker, a glial marker, GFAP was really telling that it was present in the patient-derived exosomes which were extracted from NXN5 and not from CD63. Similar proof of concept was done using SEM imaging, which shows the high density of exosomes I have on the device, proving NXN5 efficiently isolated GBM-specific exosomes in patients. Next, I'll be talking about the analysis that we performed using NTA data. So from all the post and pre-sonicated samples that we had during the chemotherapy cycles, we tried to see if there was a difference collectively in the post versus the pre-samples. Well, not really. There was no significant difference, and then we tried to understand what could be going on among the patients individually. So we looked at the exosome release dynamics with the opening of blood-brain barrier, for which we used these intraoperative samples. We calculated a ratio, pre-sonication exosome concentration upon screening exosome concentration, and we found that for patients who had a low ratio less than the average, 1.6, the peak exosomal concentration was seen at 20 minutes after opening of the blood-brain barrier, while for the other group where this ratio was high, the peak was obtained after 45 minutes, showing that blood-brain barrier opening was heterogeneous among these patients, and which could explain why collectively there was no difference in the post and the pre-samples. Next, I tried to see if the exosome concentration data correlated with MRI enhancement data that we had for the patients, and we saw that for 9 out of 17 patients, there was a pretty good correlation between the two factors. Interestingly, for one patient, this was a case of pseudoprogression. The exosome concentration showed a decrease, while the MRI showed an increase, proving in one case that the exosomes were a better predictor of pseudoprogression. However, there were many other patients for which there was no strong correlation seen, and we think that other confounding factors could be the reasons. Another interesting aspect was looking at the overall survival in these patients, and we saw that when we looked at the exosome concentration slope at post-operative day 45, we saw that one group which had a negative exosome slope showed a lower survival compared to the other group. Overall, we proved that therapeutic monitoring in GBM is challenging, and we need biomarkers like exosomes. In this study, I was able to show that we can use a microfluidic device to isolate these exosomes from patients, and theoretically opening the blood-brain barrier using ultrasound-based sonication showed some heterogeneity, and hoping that this can complement MRI in the future to make progression or monitoring of these patients easier. With that, I would like to thank everyone. I have my poster session tomorrow, poster number 20. Please stop by for questions and concerns. Thank you.
Thank you, Abaha. Do you have any questions? One question.
Hey, so in terms of focus ultrasound for getting drugs into the brain, that's worked quite well. I guess, how would you explain your hypothesis that you're expecting EV release into the circulation when there might be pressure differences from the vasculature into the brain? So you've got a gradient of molecules going into the brain, not necessarily out of the brain.
That is a very good question and a difficult question. So in terms of biology, we are really struggling to understand this, and there are many factors like one of what you pointed out. We have not looked into demarcating that zone yet, but I would like to know how we can investigate that part. This is a challenge, and right now, well, the way this is a retrospective study, and the way it was designed was collecting all the samples and seeing firstly the exosome concentration difference. Hitting that hypothesis that this could be because of the blood-brain barrier opening is, I agree, is kind of an extreme, and understanding the dynamics of that is, I think, where we will go next.
Thank you.
Please.
Yeah, terrific talk as well. It's kind of along similar lines to my question. Obviously, the blood-brain barrier exists for a reason, and opening that up can be really good for potentially allowing therapeutics to enter. How safe is it? Because obviously, with neuroinflammation being kept by the skull, letting things into the brain without the controlled exit of fluids could lead to complications. So, just a more fundamental question: like, how safe is opening up the blood-brain barrier?
in the context of GBM, anything you try to do for intervening these patients and trying to increase their lifespan by a month is safer compared to what the disease already does to the patient. So it has been proven to be safe. Like injecting microbubbles and opening it doesn't do a long-term harm to the patient. The most severe symptoms that were seen, and I cannot isolate other factors from it, were just headache and nausea in some of the patients in that cohort. But I think getting the drugs into the brain in that context, this is proven to be more beneficial than harmful.
So just to follow up to that, so you think maybe for future uses of opening up the blood-brain barrier it would be more of a last resort for really untreatable neurological diseases where there's no other way, like obviously because it comes with pretty high risks.
I would think so.
Okay.
Yeah. Thank you.
Okay, thank you. Thank you. So next speaker is Leanne Klepp, Aalborg University Hospital, Department of Clinical Immunology, and her topic is EV biomarker discovery for ultra-early differential diagnosis of stroke. Please.
Thank you. Thank you. How do I shift? Yeah, hey everybody, yes, I'm Jan. I'm a PhD student from the Department of Clinical Immunology at Aalborg University Hospital in Denmark. So a quick trip of 24 hours. Yes, and I would like to talk to you about my project, about trying to use EVs as biomarkers to distinguish stroke types for an early diagnosis already in the complex. And the reason we're looking at this issue is that worldwide there's statistics that shows that 12 million, we have 12 million strokes incidents a year, and the mortality rate is in average that someone dies of stroke every 3 minutes. So that's quite a lot. And stroke is also the second leading cause of death worldwide, and is the cause of major long-term disabilities. So today, the approach we do when we have to set a diagnosis is that we use neuroimaging. And that is a reliable method, however, it is very time-consuming, and not every hospital has this opportunity. It's expensive, and we require experts to look at these images, and that also takes time. And that means that worldwide, only 7% of those who have an acute ischemic stroke receive effective treatment within the 4.5-hour window, which exists within stroke treatment. So the project is called Stimulate, and our ambition is that we want to try and identify biomarkers on small EVs to distinguish the stroke types, acute ischemic stroke, and intracerebral hemorrhage. And for this, we would like to use to enable molecular diagnostics already in the ambulance when the patients arrive. And that we would use to minimize the weight of your stroke treatment by being able to drive the patient to the correct treatment center directly. Today, sometimes what you do is that you take the patient and drive to the nearest hospital, and then there they gotta set the diagnosis. But it's not all hospitals that have every treatment. Then you have to go back in the ambulance and then be sent somewhere else. And if you have a hemorrhage or acute ischemic stroke, this can really cause major long-term disabilities afterwards because of the wait for your treatment. So again, this is our ambition. That's why we're doing this project. And the project overview, you see up here, we started back in 2018. There was another PhD student, a doctor called Rolf Baumfeld, who has spent some years on collecting blood samples. What they did was when the patient arrived in the ambulance, they had a set of criteria which the patient has to check off, and if they did, they were included in the study. And a blood sample was drawn in the ambulance and frozen down, and the plasma was separated, and the plasma samples from the ambulance from these patients, which I have been working on, and that is the data I'm going to show you. With these samples, I analyzed them with EV array. You can compare it to the high fruit ELISA. I'm going to come back to that. To try and find and evaluate these biomarkers on the small EVs, to generate data for a prediction model. And the prediction model is what we're working on now, to try and predict the diagnosis from these biomarkers, which we're finding with the EV array. So the cohort of this project is that we have 209 patients coming into the ambulance, and of these patients we have the stroke group, which consists of 63% of the 209 patients, and 37% was the control. Control is non-vascular patients. That means that it was patients entering the ambulance. But later we found out that that diagnosis was not a stroke, it was something else, neurological, but not a stroke. So that is why we're using those as a control. And within the stroke types, you can see here we have 20% with the hemorrhage and 80% with the ischemic stroke. And that is quite common, that the hemorrhage patient just is a smaller part of these two stroke types. Well, about the method, I haven't seen anybody else's post about the EV array. I think it's a method we have developed in our lab. I can highly recommend it. Very good screening method, amongst other things. And we have been using this to detect the biomarkers in these patient plasma samples. We have been targeting 52 stroke-specific biomarkers, which we have found in the literature associated to the two different stroke types. We have made a panel, and this panel we have. Can I use this? Oh, no, sorry. What is the try point? Step one in the EV array is that we print the antibody targeting the biomarker of interest. The biomarker of interest will be a protein in the membrane on the outside of the EV. We do not look at anything inside the EVs, only in the membrane on the outside. Then we would, in step two, we would add our sample onto our printed antibodies. It would capture. We capture the EVs. This is the preliminary results. I have a non-disclosure agreement, so I can't show you more. But we could see with our EV array, our data, that we got some significant markers out that could indicate that they could be used for discriminating between the different conditions. And thank you for raising that item.
Any question?
Hi, thank you so much. Great talk, guys. I wonder, guys, what kinds of stroke included, like some dissection or like some other type of stroke type, like lacuna or atherosclerosis or cardioembolic.
I'm just going to repeat so understand what you said.
I'm wondering what kinds of like some ischemic stroke are included, like lacuna stroke or the atherosclerosis or the cardioembolic, and also the kind of layer arterial dissections, also the kinds of issue in the clinical setting to make the diagnosis. So that's
Okay, that's also a good question because we didn't describe that here. In the inclusion of these patients, it has been all that has a blockage. So we haven't distinguished the subtypes. They're very roughly divided. Yeah.
Thank you so much.
You're welcome.
I love the idea of collecting samples in the back of an ambulance. And so my question sort of follows on this in terms of rapid diagnostics, right? You're currently using a platform. What's the time frame and what's the sensitivity that you can work with that EV capture array that you're using? And do you maybe envisage being able to translate that to being able to diagnose what type is a stroke or a hemorrhage in the brain in the ambulance in real time?
That's also a good question because I didn't get to talk so much about that. This is in the investigation part to find the biomarkers. We use EUV array, and that takes 2 days, depending on the amount of sample you have. Of course, we don't have 2 days in the ambulance, so next that follows, when we have found our biomarkers, then we're going to do the mail detection method. We are working on that now. I have a non-disclosure, so I can't say so much about that part. But yeah, it has to be very sensitive because we're looking at EVs that has to travel with the bloodstream from the brain outside and down. So of course, it has to be a very sensitive method. And we have one which we think will work. So for the next year, I'll let you know.
All right.
Okay, so thank you.
Yeah, thank you.
We will go to the next phrase. I'd like to introduce our Professor Yin Zhu to share the next phrase. Okay, please.
So our next speaker is Gina Ko from University of Pennsylvania, and she will be talking about double digital assay for single EV and single molecule detection.
Hello everyone, I'm Gina Ko from the United States, from UPenn. So today I'm going to tell you a little bit about single EV, but also single molecule profiling technologies. So our lab's working on developing different next generation tools for medical diagnostics, and because we're very interested in diagnostics, we are very interested in trying to develop technologies to think about how to profile EV better and also with more sensitivity. So there are multiple technologies that we develop, and then we're trying to make it high throughput, ultra sensitivity, and multiplexing. And for today's talk, I'll tell you a little bit about the single EV technologies and also the single EV and molecule technologies. I'm sure everyone's familiar with what EV is. We are very interested in EVs because they can be really good diagnostic but also therapeutics tool. But then the problem is that they're very small, there's high background because all different EVs are shed from all different types of cells and also very heterogeneous. So to solve those challenges, we were thinking about doing single EV profiling. If they're very different from each other, why don't we just separate them into a different chamber so that we can just pick up the chamber that has EVs in there so that we can solve the needle in a haystack problem. So our first approach was doing single EV droplet digital PCR and sequencing so that we can use droplets as a chamber to separate individual EVs. We first started with droplet digital PCR, but then we actually ran into some issues of low throughput because it's more like imaging based, and also low multiplexing as well because it's limited by the number of fluorophores that you can use for imaging using fluorescent microscopy. That's why we scaled this up to sequencing so that we can profile more EV and also more biomarkers. So the single EV droplet digital PCR technology pipeline is as follows. You first start with EVs isolated from different cell lines or different plasma, and then you label the EV with antibody DNA because we're interested on the membrane protein on the surface of the EV. And then you use the DNA so that we can use it for amplification, but also for barcoding, so that we can do multiplexing. And then we encapsulate that into droplets, and then we do thermal cycling for droplet PCR. Here's a droplet PCR generator where you can mix different master mix of the PCR. And then here we actually visualize using bits because we all know that EVs are really too small to be visualized. But then we use the same calculation so that we can add individual EV into the droplet using the Poisson's distribution so that we can achieve single EV loading. And then we move on to single EV protein sequencing, meaning that we're profiling proteins on the surface of the EV, but then the readout is sequencing because we're using the antibody DNA conjugate that I showed you before. So here the pipelines start with EV, we label them with antibody DNA, and then we encapsulate them inside the droplets. As you can see in the video, they're actually pretty similar to single-cell RNA sequencing, where you add barcoded beads inside the droplet with single cells, in this case single EV, and then the sequencing. But it wasn't that easy, because when you think about EV versus cell, EVs are at least 100 times smaller than the cell, meaning that the volume-wise, the content that you're going to get from EV will be extremely small. So we were not able to use the pipeline that was optimized for single cells. So we actually developed a sequencing protocol for single EV profiling. So the first step is extension in droplets. So inside the droplet, you have barcoded bead that has EV barcode, UMI, unique molecular identifier, so that you can count the number of molecules, and then the A, so that you can hybridize the sequence to A prime on the DNA antibody conjugate. That antibody DNA conjugate also has antibody barcode, so that you can do multiplexing. It also has T7 promoter sequence, so that we can do the second step, which is IVT, in vitro transcription. We perform IVT because of two different purposes. One, we want to amplify the signal as much as possible so that we can get EV readout from single particle level, but also we want to remove any background so that we can increase the signal to noise ratio. So from the DNA products, we did IVT so that we can produce a lot of different RNAs, and then unamplified or somewhat amplified DNA that is not complete we're going to be degraded using DNase treatment, so that we can purify RNA and then do RT and PCR and perform sequencing. So we first compared with bulk EV measurements and also single EV, no EV as a control, and you can see good amplification for all different samples, and then you can see that the control amplification was a lot later. And then for bulk versus single, you were able to see the right band from the gel. And we also did Sanger sequencing, and we were able to find the actual amplicon that we were interested in. So using this technology as a proof of concept, we were able to profile ROD264 macrophage cell lines, individual EV profiling and their expression from all different proteins that we're interested in. And we were able to get different subpopulations as a composition, which can only be done when you're doing the single EV analysis compared to bulk EV analysis. But then when we are working on single EV analysis, I'm pretty sure a lot of people will going to agree, if you also work on the single EV analysis, where even though you develop a platform, you're going to lose a lot of data because the actual inherent biological amounts coming from single EV is really, really rare. So we were thinking we actually need a new technology that can not only look at individual EV, but also we can count individual molecules coming from single EV so that we can make it ultra high sensitive and actually measure all different individual EV composition. So here we turn the new technology called double digital assay, where we look at individual EV and also individual molecules from individual EV. So this work was done by my PhD student, David Reynolds. So he actually made a microfluidic system where he has a honeycomb-sized wells, and then that is coated with a monolayer of microbeads, so that we can achieve double compartmentalization. So each well can serve a chamber so that you can put individual EV into the chamber. But then when the EV is inside the chamber, what you can do is that you can lyse the EV so that you can get the material from the EV to the chamber. And then those monolayer of the microbeads can capture those molecules, but then we make it so that only one bead can capture one molecule so that we can perform digital quantification. And in order to get readout from single molecules, we perform TRMA signal amplification, TSA, so that we can visualize individual beads coming from individual chamber. So we perform TSA, TRMA signal amplification, first from cells, and then we compare TSA versus no TSA to see how much amplification that we can achieve. And also we apply to EVs, TSA versus no TSA, and then you can see that only from the TSA sample we're able to get some signal. We perform single particle loading so that we can add only one EV per well, that micro well. And as you all know, EVs are really small. That's why we use cells as single particle loading, and then we use the same calculation and Poisson's distribution to do single EV loading. But then the reviewer of the paper said that they really want to see the EV inside the well. That's why we will perform this test again. So here we labeled EVs with calcium green so that we can visualize them. And then we loaded them into the wells with the same calculation that I briefly mentioned, and we were able to achieve single EV loading. So we profiled PD-L1 expression from EV, and then we compared bulk versus single EV. So we prepared two different cell lines. One is PD-L1 knockout cell line, melanoma, MEL264, so that we do not see any PD-L1 expression. And then the other one was PD-L1 positive cell line EV, so that we can compare those two as positive and negative. So from the bulk EV, you can see that when we do off-chip bulk EV measurement, there's no signal coming from PD-L1 negative, there's some signal from PD-L1 positive, and we're able to recapitulate the data also using single EV analysis platform using our technology. So as a final data, we were able to show that we can actually count the number of molecules coming from single EV so that we can quantify the PD-L1 expression from individual EV. And we wanted to compare that to the true population and expression. So what we did here was that we performed ELISA using known PD-L1 molecule concentration, the PD-L1 protein, and then we made a standard curve. We looked at the PD-L1 negative and positive cell-derived EV and looked at the actual average PD-L1 molecule expression coming from those two different EVs. We also performed, using our technology, single EV, and then we show that the actual number of PD-L1 molecules per EV actually matched to the conventional technology, which was around 3 to 4 molecules per EV. And we also showed that the limit of detection of our technology was 40 times better than conventional ELISA. But because we can actually look at single EV, not on bulk, what we can do is that we can actually find the distribution of different EV populations with their expression level of PD-L1 molecule, as you can see in the histogram, where we can look at different positive number of bits and all different number of EVs as well. So for future directions of this technology, we are actually working on Alzheimer's disease diagnostics. So we're trying to find brain-derived EVs coming from plasma and CSF and try to look for different brain markers so that we can identify different diagnostic patterns. And we're also working on different single molecule detection technology development. With that, I really want to acknowledge our collaborators, funding source, and amazing people in our lab. Thank you so much.
Thanks, Gina, for a very nice work. Very good demonstration of single EV analysis that I mentioned yesterday. Due to the time limits, just one burning question.
Okay, thank you. Very nice talk. I have a question how many West you can count through droplet or the chip? Because if you're using the, if you have to make them single, you have the many droplet and many well. And but the EV in the plasma or fluid, they are very high concentration. Then you mean you need a lot of the a very big chip or a large, a very long term to treat the sample. Yeah,
yeah, that's a really good question. So for droplets, so it's a matter of like how long you run the chip so that you can produce a lot of droplets. We usually make around 1,000 droplets per second per chip, but then you can always parallelize the chip up to like 10 chips, 20 chips, so that you can increase the throughput. In terms of the wells, it's a little tricky because it's physically there, right? Here we only use 10,000 wells, which means that we only looked at 1,000 EVs, but they're very small, more like millimeter by millimeter size, so you can scale up, but there will be some limitations.
Okay, thank you.
Thanks. Let's thank Gina again. If you have more questions, please feel free to ask her later. Our next speaker, I hope I pronounced name right, Kiro Aslam from University of Turku, who will be presenting high-sensitive rapid detection of urinary EVs with upconversioning nanoparticle-based lateral flow assay.
Hello everyone, my name is Kiro. I'm working at the University of Turku, Finland, with collaboration with Inogen company. I'm a postdoc over there. So today I'm going to talk about lateral flow or rapid test development for the detection of urinary EVs. So of course our target is diagnostics, and as a case study we used bladder cancer. So you all know about why we do the bladder cancer, because urine has direct contact with bladder cancer and it sits there for a long time, and there is a huge chance of urinary EVs over there and with the different kinds of cancers states. So why we took the bladder cancer, there is various reasons, and there is a global issues with, there are many people who are diagnosed and are dying due to the bladder cancer. And currently, the problem is with the bladder cancer is this is highly recurrent. About 70 to 75% patients are recurrent within 5 years. And that's why the diagnosis of bladder cancer is highly costly compared to any other cancer due to the recurrent rate is very high. Yeah, so the current test on the market is cytoscopy and cytology, but they have the problem of sensitivity and specificity, and of course the invasiveness. And there is no biomarker test currently in the clinics. So that's why we need a highly sensitive and really time-consuming overcome test that would be specific and sensitive, and of course non-invasive and simple and low cost. So that's why we think about the lateral flow immunoassay, and it's the immunochromatographic assay, which is well established in the point of care diagnostic, and you all know about the advantage is the real-time results it gives usually. Result is within 1 hour, as like as the pregnancy test or COVID test kit. So you know already that's how we should read those COVID test kit or pregnancy kit test is the same one. So which is negative when is the control is red line, there is no test line. The positive is when is control is positive and test is positive, and the invalidity when test is positive and there is no control, and the invalidity is there is no test line or control line. So now how we build those test kits. Here is the methods and materials. We have the nitrocellulose membrane and the sample pad and the baking card and wicking pad. All together we laminated into one card. And then we print our antibody in case of tetraspinin with the dispenser. So then we cut our card as a strip of 4.8 millimeter with the biodot cutter, and then we measure our strip with Upcon reader that came from Laprox Inogen company. So here is the overall workflow that I would like to share with you. So first we remove the cell and cell debris with 2000 G for 5 minutes, and then we add those 30 microliter of urine sample with our Upcon nanoparticle coated CD63 antibody on the 96 well plate, and then we add those sample after 40 minutes to the strip where test line and control line exist. You can see the test line and control line, how it works. The test line has the bottom has the CD63 antibody as a capture. These CD63 capture EVs. Those EVs go back with the detection of same CD63 antibody that is coated on up-converting nanoparticle. And on the right side is the control line where same settings is present except the EVs. And then after 30 minutes incubation, we take the reading with the UpCon reader and we get the quantitative signal. So one of the benefits of up-converting nanoparticle is there's the photostability and autofluorescence is really low, and the narrow emission spectrum. And what is up-converting nanoparticles is it exhibits photon conversions, and it involves the lower energy to the higher energy. And how it works with the emission is 55 nanometer and the excitation is 98 nanometer. So to validate our assays, so we have used different kind of samples. Of course, we use the isolated EVs, that is filtered, and also SEC-based EVs, and also ultra-sensitivity derived EVs to prove our lateral flow assay can detect EVs. And then, as I said, our target is diagnostic, that's why we use the clinical sample. So here's the limit of detections of the CD63, CD63 up-converting nanoparticles. Here you can see we can detect the EVs quite nicely with the ultra-sensitivity derived EVs. Here is the different isolation techniques that we have used, just make it prove that our assay capture and tracer is specific for the EVs. So the first three graph A, B, C from the urine donor 1, urine donor 2, urine donor 3, that we have used the filtered EV, where EV is removed with the filter. Then you can see less signal. When we concentrate with the filter, the EVs, then we get more signal. When we put the EV as it is, then we see the normal signal. And the same happened with the urine donor 2 and the urine donor 3. So it was the healthy urine donor. And what about the cell culture medium that came from one of the cancer cell lines, so in the D. So it's the same thing happened with when we remove the EVs, there is almost no signal. When we concentrate more EVs there are more signal. When we use the cell culture medium as it is, then we see the normal signal. So what is the linearity of the EVs when we use the SEC isolated EVs? Then we use 2 concentration, 500 nanogram and 1000 nanogram. We see the linearity that detect the SEC EVs. Then we also isolate EV from the pool sample of bladder cancer sample and benign prostate hyperplasia, that is we used as a benign. So we use the 2 fraction, EP fraction and other protein enriched fraction, and you can see in the last figure F. So the more signal is coming from EP fraction compared to the other protein enriched fraction. So as we know that our assay setting is working quite nicely with isolated EVs, then as a diagnostic purpose, so we use directly the clinical sample here. Here we use 30 microliter EV sample and we remove the cell and cell debris with 2000 G for 5 minutes. And you can see the results that the red one is the bladder cancer, 62 sample. That is quite nicely, significantly separating benign prostate hyperplasia, that is we used as a benign. This is clinically challenging group because it's the comorbidities as near as possible that we have. And of course we have the healthy, that is 15 to 16 times higher signal that coming from cancer side compared to the healthy. The one note here is the healthy was the young from our department. So it's not the age matched, but the benign and the bladder cancer was the age matched, and then the same procedure applied for the collection. So about my last slide, so the conclusion is the up-converting nanoparticle assay platform suggests that the feasibility of tetraspinin assay can enhance the EV detection that can facilitate the bladder cancer more specifically. And we have also working with in the future directions. So we have the up-converting nanoparticle to different fluorescence. So then we can detect the dual epitope in the same EVs. So you can see from the figure that the test line and the control line have the dual signal. So then if we combine, then you will get the more simultaneous detection in the single test line. So another future aim that we have been also working with the anti-glycan specific detection of EVs, where we'll capture EVs with the tetraspinin and other cancer-associated marker that is already published and known for the EV specific, and go back with the detection with the anti-glycan specific lectin or anti-glycan specific antibody. That's all. Thank you very much.
Thanks, Kiro, for keep our time back on track. We have time for a couple of questions. Carlos?
Yeah, thank you for the presentation. That was great. I just wondering if you did some analysis of sensitivity and specificity in your data. I think you mentioned the rationale. One of the reasons that you're trying to do this is because whatever is in the market is not sensitive enough or is not specific enough, right? And I think this is a message probably for all the speakers or for everyone, right? If you're gonna be in the biomarker business, you have to demonstrate that your system have enough sensitivity and specificity to be implemented in a clinical practice or in the healthcare system, right? And if you're looking for example, a cancer for high-risk population, you need to show enough sensitivity, right? Able to identify the true positive in your population. If you are pitching this as a more screening program, the specificity is what you need to take care. So be able to identify the true negative in your screening program. So I think, I don't know if you did, but you showed the difference. So different doesn't mean anything. You have to show the sensitivity and specificity of whatever you are checking in the biomarkers.
Yeah, thanks for the comments. Yeah, you're right. So we are going that direction. So first we develop the protocol with the tetrasprin, otherwise people will not believe it's the EVs. So we are still in the development phase. So the next we have really some good data on it, but we are still proving and we are going that direction.
Yeah, hi. Just wondering, how do you know whether the EVs are specific against bladder cells and not related or produced by kidney cells exfoliated in the urine? Is there a way to separate those?
Yeah, you asked a very good questions. It could become also from prostate, also from kidney, but it was the bladder cancer specific sample, and the benign sample was also from the comorbidities of the benign diseases. So as the clinician, it marked with the bladder cancer, so that's our belief was the EVs might be from the bladder cancer. But it could be also from prostate cancer, it could be also from kidney cancer.
Thank you so much. I'm from Japanese Foundation on Cancer Research. I'm also studying the urine EVs. So my simple question is about the concentration of the urine EV. So as you know, the concentration of the urine is quite different among during a day, the morning urine or evening urine. So how did you normalize the urine concentration in your assay?
Yeah, very good question. We didn't do the normalizations for these assays, but we also when we really run the cancer-associated marker or the cancer-specific marker, then we also do the normalizations with the tetraspinin marker. Then we do that value for the normalizations. And we do also normalize the creatinine values and other values. That helps us to get into there.
Let's thank you again. If you have more questions, maybe ask me later.
Oh, okay.
Yeah. Thank you very much. So our last speaker for this session is here. Yeah. Vasily Chanoshev from National Medical Research Center for Obstetric, Gynecology, and Perinatology from Moscow.
Yes, thank you very much, and thank you again for allowing me to present our work. Now, one of the issues is, you probably know, that received quite a lot of attention in recent years is, I mean, we have a lot of progress in technology, right? I mean, next generation sequencing, omics, etc. However, when we think of actual use of this, especially in rural areas, and for quick testing, of course, its expense and tediousness and of work required to perform analysis is kind of one of the drawbacks, and it's been brought up in recent publications in high impact factor journals. So one solution could be, for instance, speaking of rural areas, I mean, we can put this large equipment and we can transport it and perform potential analysis in areas further away from hospitals. However, as you can imagine, it still might not be the best solution, although it's been used quite a lot recently. Now, another solution is trying to miniaturize biosensors. So this example is just one of the many kind of publications in this topic is where, for example, performing analysis in a small chip. For example, CRISPR-Cas was integrated into a small chip, and analysis can be done pretty quickly without using standard equipment. So what we kind of came up with, and what was our idea, is try to combine photonic integrated circuits and microfluidic devices. Well, as you might have heard of, photonic integrated circuits, which is expected to be in the near future integrated into just computers and iPhones, is where we're not dealing with electrons anymore that have kind of some disadvantages. For example, it essentially can go into heat. So, for example, where computers and iPhones always need cooling. In this case, we're working with photons, and it allows, in fact, to transfer for saving information, etc. So the interest in photonic circuits emerged quite rapidly. Well, our idea was to kind of combine several areas, and that is including microfluidics, microheaters, we can locally control temperature in our chip, photonic integrated circuits, and of course surface modification, the chemistry that is all behind making the surface activated in order for us to capture what we want and perform analysis. So what we started with is Mach-Zehnder interferometer. I mean, there's a lot of photonic integrated circuits that we can come up with. In reality, you can come up with something new yourself today, in fact. So there's really an infinite amount of type of circuits that you can come up with. Well, Mach-Zehnder interferometer is one that is well known. And what we have is basically light entering interferometers, and it becomes split into a sensing and reference arms. And after which the arms are recombined in a wide junction, and the interference is measured in the output. So, and basically we can quantify the presence of the analyte that was deposited on in the sensing arm on the surface. So we're basically combining this type of photonic chip with microfluidics. Microfluidics, I mean, we've been working mostly with just polydimethyl siloxane, PDMS, and making this kind of device, which of course is still experimental. It's easy to disassemble in order to perform different measurements. So the idea is, well, we take this microfluidic device where we have a channel roughly 300 micron width and about 50 micron height, covering the sensing part of the Mach-Zehnder interferometer. So what we first first need to do, as you can imagine, we need to modify the surface somehow we need to activate it in order to capture the vesicles that we want. And perform washing, obviously, and then look at how the resonance wavelength shifts. So it's kind of like indirect measurement. And that way we can actually perform quantification. So the first thing that came to mind, well, what are we going to modify the surface with? So there are several molecules that we can use in order to capture. So antibodies are very commonly used. However, in the recent years, DARPin and aptamers, which are synthesized and do not require animals, have received a lot of attention because especially DARPins can have very high specificity, in fact better than antibodies. And we've decided to use, take a look at two well-known markers, HER2, which is very known in breast cancer, and EpCAM, another marker very typical in vesicle research. So, so the first thing, of course, was to obtain EVs, and a nice kind of ideal model is where we have two cell lines, one of which actually expresses HER2 positive vesicles, and the other nearly none. And these two cell lines are what we decided to use. We confirmed by Western blotting that, in fact, SCO3 is more enriched in HER2 marker. And here, in this case, we decided to use DARPins, as I mentioned, in order to modify the surface, activate the surface. And the general kind of idea was to use this relatively standard and well-known method of attaching the molecule that we want to capture vesicles with by using APTES and EDC and HS carbodiamide cross-linking. And the tunable laser that actually is the generator of our, what we're generating our photons with, and that are actually transported through the photonic circuit. So the first thing, of course, we need to activate the surface of the chip, which is silicon nitride, and that can be done by plasma treatment. So we obtain OH groups, and APTES is a good linker that we can later use to attach our DARPin. So we eventually obtain a siliconized surface. So then we need to have some molecules that will actually allow us to attach DARPin. And like I mentioned, this is the EDC and HS approach. So finally, what we have is a reactive sulfone HS ester and a siliconized surface, which eventually would give us a covalent linkage between the surface of our chip and the molecule and DARPin. So here's what we've done. So here in the y-axis you see the relative deep wavelength shift that I've been talking about with this indirect measurement and protocol time. So first, what we're starting with basically is again a prepared silicon nitride surface, so that we've modified with APTES. And now what we have flowing through the channels is just buffer. So you can see there's pretty much no change. Now we introduce the DARPins that was modified in order to covalently link it to the chip, and we can clearly see quite a significant shift in the deep wavelength. Then we perform washing. Of course, we want to get rid of any DARPin that's left, and then we introduce the vesicles, and again we have a significant shift. And then we do another wash in order to remove any vesicles that did not bond or were just in the solution. So eventually what we've got is actually interesting results. So what we actually wanted to see is we can see that the vesicles that are expressed by the MDNE231 cell line, there was a significantly less shift in the deep wavelength, which is what we expected to see. And so this is kind of like a proof of concept in the fact that this modification and activation of the surface, we can use this as a really nice sensitive sensor. And to confirm that we in fact captured vesicles, we've done scanning electron microscopy, and we did see that on the active zone of the photonic integrated circuits, we could see the kind of the ring-shaped vesicles. Now, this is one approach. Another approach, we thought, well, let's try also to use similar cross-linking, but with strep-dividing biotin. So in this case, we decided to use plasma vesicles that were, again, isolated by SEC, similar to the case previously. And the modification process of the surface was similar. So here we again analyzed the surface, but in this case, instead of the dark green, we have strep-dividin that we want to attach to the surface. Now, after we do that, we can use biotinolized optimer, for instance, and this is what we decided to try in this case. So eventually, we get again a chip ready for EV capture. Again, this is covalently bond strep-dividin, and then the strep-dividin biotin interaction. So here we tried to use a different concentration, and one thing I didn't mention is that the optimers that we used were fluorescently labeled, because this time we actually wanted to see, well, can we fluorescently check that we're in fact modifying the surface, how uniform it is, etc. So we could, so we saw by fluorescent 3D tomography that we have a pretty much equal modification of with CD63 optimer. So we have this is the first optimer that we used for decided to use for capturing vesicles, and you can see that in each small microchannel, short microchannel, we have pretty much the same nearly the same intensity. Now for testing, we used 3 different concentration of vesicles. And for one microchannel, we decided to not introduce vesicles at all, but still introduce the second optimer to see if there's any non-specific binding. And what we saw from fluorescence is that pretty everything was logical. So with increasing concentration, we saw a more intense signal, and where we did not introduce EVs but introduced a secondary optimer, we saw nothing. So, which was a good sign that there was no non-specific binding, basically, of the second optimer on the surface. So this is another approach that we tested, and it in fact was successful. However, just to kind of summarize this kind of presentation, is that we've shown you that this photonic integrated circuits with microfluidics can, in fact, be used to make a platform for EV identification, pretty much any EV identification. So the eventual design, of course, right now it's experimental, it's kind of R&D. We have not yet miniaturized this whole system, but it's not difficult to do. So of course, the end product. The end result would be a small kind of chip, where the microfluidic channel would be the only thing that we're changing in the system. But a lot of work is still yet to be done, because we would like to, of course, transition to more realistic samples, not just vesicles that were isolated from human blood, for example, but also test and see if we can actually, without isolating vesicles, actually capture them. So that would be the best scenario. And of course, we can test other photonic integrated circuits, and our kind of near future project is we're collaborating with a hospital where we would like to use the system in order to evaluate the effect of treatment on the expression of HER2 marker, which is a well-known marker in the breast cancer area. So I would like to, of course, thank the collaborators. It's 4 main organizations that were involved in this, and each one is like a Ford, model. So each one is specializing in its own field, and without their contribution, it would be difficult to actually get this project going. So I would like to really thank, and of course, funding the Russian Science Foundation for allowing us, giving us financial support. Thank you very much.
Thanks, Vasily, for the work. Any questions? We have time for one question. No? Okay, I'll ask the question. How do you think you solve the non-specific binding issue?
Yes, it's a good question because we, in reality, we still, as you know, we're one of the common ways to kind of cover the unmodified surface with the CA, right? And we did not do that. And in fact, this our hypothesis is that we of course are using, when we're modifying the surface, we always use excess of the what we're modifying the width. So it appears that there might be nearly complete coverage, so the surface becomes completely modified with it, which kind of helps to pretty much avoid non-specific binding. But we still would like to have a more complete perform complete testing. So in this case, the test that I've shown you, we did not introduce vesicles to see if there would be non-specific binding of the second aptamer to the surface. So we showed that works, but there still needs to be more references used in order to really show that, in fact, maybe we don't even need to use this BSA in order to solve this non-specificity issue. So.
Thank you very much. I think that's the end of our session. Let's thank Vasily and